Well, Dr. Muriana, thank you so much for that nice introduction. It really sounds rosy when you graduate from an excellent facility like FAPSI and get into academics, which you really want to do from the day one that you joined here. Many of you I know that you are sitting here maybe just as an audience, not a student, a staff, or a faculty. But I was once sitting just like that. My wife said, there's a research symposium, you want to come and attend? And I did. And a lot of the work the people presented really got me interested in that. And that's how I led into Dr. Muriana's lab. But anyway, I hope you'll also get interested once you spend your time today. If anybody can't hear me, I can turn on the mic. Vinita, can you hear me? I think she can't hear me. Okay, without much waste of time, let me start with my introduction. Dr. Muriana already introduced me, but I will just talk one or two slides about the place where I came from. This is the title of my presentation, Food Safety Challenges. I kept it very broad so that it can be applicable to a broader audience. University of Louisiana Monroe is the place which I had never heard of before I actually accepted the position. It's not a very big university, but it's a very dynamic university. They're doing a lot of new things, and they have a very good program in pharmacy. There are only 5 or 6 toxicology programs in the US who offer undergraduate degree in toxicology, and these folks are one of them. And they are branching into food safety and food toxicology research, and that's how I got my position there. I joined there in end of 2012, and since then I've been there. If anybody has not heard this place, Louisiana comes, the University of Louisiana Monroe comes somewhere very north side of Louisiana, right across the border of Arkansas. And even you can drive from Oklahoma, but it's about 8-hour drive, which we did. And this is a part of University of Louisiana system, which is a group of 9 different universities and colleges. With that, let me start with my talk. Okay. Today what I'm going to talk about is a very general and very basic information about some of the food safety challenges. And there are a lot of food safety challenges in most of the disciplines in food safety area. There are challenges in food engineering, food chemistry, product development, so many other things. But my talk will focus mainly on food microbiology and food safety in that issue. I will start with some of the food safety impacts. That means what are the what it means for a human health, and then I will get into how the foodborne illnesses are investigated in the US, what is the system they follow, and what is the exact estimate of illness burden in the country, and how serious it is, the food safety challenges. And I will also deal with some of the issues of emerging food pathogens and the challenges that we are facing, that we have been facing since from 20 years actually. And if the time permits, I will talk some of my work that I did after moving out from here. Let me start off with a very simple basic definition of food safety. Food safety is an easy word to define, but a difficult word to understand because it's a big, broad, umbrella-like term. For many of the people, food safety is just like a food hygiene. We call anything that you do in to make a food safe, whether it is washing your hand, processing, or cutting, chopping onion, anything that you do in a safely manner, that is a food safety practice. And there are pretty standard ways of how to avoid foodborne illnesses that are called as clean, separate, chill, and cook. These are the 4 easy ways how you can avoid foodborne illnesses. We will not get into that, but it's very easy to understand that you have to keep the food cold, cold food cold, hot food hot, if you don't want to get any contamination in day-to-day activities. And again, food safety has no standard definition, but it is the state of acceptable and tolerable risk of illness, whether it is a hazard or any health issue. If it is tolerable and acceptable in the sense from food industry or regulation wise, if it's tolerable from your human body, that if you're able to not fall sick for a long time, that means you are consumed a safe food. Since we are all here, that means we have been eating good safe food since 2-3 days, I guess. So food safety has both acute and chronic kind of impacts on human health. Acute in the sense we talk about very short-term impacts like vomiting or food poisoning or diarrhea, these kind of things, which will go off in 2 or 3 days. But if it continues, the foodborne illness, if it continues, it leads to long-term impacts on your health. Some of those can come from consuming a very nutritionally inferior kind of food. For example, I know many students survive on the noodles just because they don't want to cook or they don't have time to cook. But if you continue to do that till your graduation time, then you'll definitely get some of the problems. That is again a food safety issue. And long-term effects include diseases like obesity, heart attack, diabetes, and so many things. But CDC, FDA estimates that nearly 2 to 3% of these foodborne illnesses lead to a long-term effect on human health. That's a big number if you know the impact on how many people get foodborne illnesses every year. What are those things that usually cause foodborne illnesses? Everybody knows that foodborne illnesses come from microorganisms, that if they contaminate the food that we eat, or the chemicals or the toxins that they are present in the food. So that is a pretty obvious thing, but it's a very difficult thing to understand for many of the people, like farmers, if they don't have any food safety knowledge. And we will talk about some of the things that are mainly microbiological origin. It includes bacteria, viruses, fungi, parasites, and many other things. And many people learn about food safety or food safety problems through the news media. Many people want to be famous, but you don't want your name to be in one of these papers, especially if it is talking about a foodborne illness. So this is how we learn, but this is the stage when a lot of things have already happened. Many of things have already gone out of control when it comes as a news in the media. It takes a long effort, it takes a long-term effort of many of the health officials to detect an outbreak. Here, whatever we read is called as an outbreak. What is an outbreak is when 2 or more people fall sick by consuming the same food and they have the same kind of illness, that's called as an outbreak. But there is an exception that not all people who, it has to be 2 or more. For example, if you come in contact with a poison like Clostridium botulinum toxin, even if there is one case in the country, it is an outbreak. So that is a pretty serious toxin. You don't want ever to come across with that. And the reason why public health officials are so vigilant on investigating foodborne illnesses because they want to keep it in check, and they don't want anything to happen for all the general public health. And they also want to learn how to meet these challenges, how to control these foodborne illnesses. That's the reason why investigation is a big step and a very important step. Foodborne disease is a very common reason why people fall sick and why people seek medical care. And many of the foodborne illnesses are not reported. I'm sure many of you must have got a stomach upset, but you didn't go for doctor many times, right? So because it was a short-term effect. But there are some problems which are very serious problems in food safety area, and then it will lead to an hospitalization, and then it will be reported to the health officials. I will just go over how that thing gets into an outbreak. But one thing is that it's a very time-consuming process. The reason why it takes so much time is, for example, imagine a scenario where a patient becomes ill after consumption of a contaminated food, and then he seeks a medical care. So it will take about 3 to 4 days for the patient to become ill so that he can go to a doctor. Within just having an episode of upset stomach, we don't normally go to the doctors, right? So if it continues for 3 to 4 days, you will end up becoming sick and then report yourself to the doctor. And then if doctors suspect you are infected with a foodborne hazard, then they'll take a stool sample and send you a sample for analysis. That will again take 1 to 5 days time, anytime, depends where your location is and how far is the health safety lab is and all that. And then it takes another long process to identify what is the exact organism that is causing the disease. That's again a time of 1 to 3 days of time. So once they identify that, if it is, let's say if it is an E. coli that got into your system, and then they have to report it to the public health lab, which will take again These days it is much faster. They might just email or send it over the internet. So then it takes again 1 to 4 days to confirm the case. Basically, they have to go through the molecular identification and things like that. So in all, food safety outbreak declaration might take at least 3 to 15 days of time. So it's a long-term process. And whenever you want to know about food safety, who does all this kind of investigation, one thing that should come to your mind is the CDC. That is Centers for Disease Control and Prevention. These are the folks. They have a system which is called as Foodborne Outbreak Detection System. They operate nationwide. I will just go over how those people come and identify an outbreak. How do these people alert the public so that public will not get infections? There are three basic things, the three important steps in this system, which is called as Foodborne Disease Outbreak Surveillance System. It is a surveillance system. First is the local health department, then state health department, and the federal health agencies. When a person becomes sick, they go to the physician, and the physician detects any foodborne illness, and then they report it to the local health department. That is your local health department. They generate your reports like healthcare worker, HCW, and confidential morbidity reports. These are the reports they are generated and send it to the state health department, who again further investigation and identification using techniques like pulse field gel electrophoresis molecular techniques. They exactly identify what is this pathogen that is causing the illness. And then they report it to the federal health agencies like CDCs and other regulatory agencies, who run the data through PulseNet and FoodNet systems, then declare an outbreak. What is a PulseNet and FoodNet system? If you want to learn more about PulseNet, you just have to Google PulseNet. It will take you to the CDC website. But it is just a network of many of the laboratories in the country. And they basically do a thorough investigation. When you say PulseNet, it's mainly laboratory-oriented research. FoodNet is population-based survey kind of thing. So they take an isolate of the organism, identify in a molecular fingerprinting method, and then compare it with so many other isolates in their database and declare this is the particular pathogen. They do this for these organisms. These are the organisms which are watched regularly by CDC. There are so many other pathogens that cause diseases, but they are not watched. For example, norovirus. Norovirus is very common foodborne illness causing organism, but unfortunately CDC is not watching that. These are the major pathogens that are being watched. And in case of FoodNet, which is again operates with the CDC, it is a disease active surveillance network system. There are 10 health departments all over the country. They regularly monitor the data and then keep talking to each other and work under the CDC headlines. But that's how the investigation background happens before you see anything that's happening in the news. But let's come and see what is the burden of illness, what is the seriousness of foodborne illness in the country. CDC again did a very important work in 2011. Till 2011, all we knew is that every year there is 5,000 people die because of foodborne illnesses. But in 2011, Dr. Elaine Scallon out of University of Georgia, he was hired by CDC to investigate foodborne illnesses burden in the country. She took a large database of 10-year data and then came up with the numbers. Now it is pretty standard. Any food safety papers that you read, these are the numbers you will find. They say nearly 48 million illnesses occur every year in the country. And out of that, nearly 128,000 people are hospitalized, and nearly 3,000 people die of foodborne illness every year. But this doesn't tell us much. We really want to know, as a researcher, we want to know what are those pathogens that are causing this, and is there anything we can do about it. Here, when they broke down these numbers, they came up with these numbers. They say that nearly 10 million illnesses are caused by pathogens that we already know about, like E. coli, Salmonella, Campylobacter, and so many other pathogens that we already know how do they behave, how to control them, how do they exist in the environment, and things like that. That's about 1,350 deaths that caused by these organisms. But the biggest problem is here, which is close to 39 million of people or illnesses that are happening. We don't know anything. They might be causing by the same organism that we already know about, but it's an entirely new system. It may be entirely new food system, or it may be entirely new host. There's a lot of things we need to learn about these many illnesses. So that's a big challenge. That's one of the big challenges of food safety research. And that is again a significant number because it's causing more deaths than the pathogen that we actually know about. But this comes to a question: what are those foods? If we know that this particular food is always is a problem to deal with this particular bacteria, then we can easily monitor that and control it. But we know that only only these pathogens that are again have been studied extensively, and we know that, for example, Campylobacter is always associated with poultry and poultry products. Salmonella, we know eggs, peanut butter, which is recent. But the thing is, these pathogens are very active and they know how to manipulate their own challenges. They can come into many other foods that are not listed here. What are those foods that are usually implicated in foodborne illnesses? You can see here, poultry, leafy greens, and beef and fruit nuts, they are causing majority of the foodborne illnesses in the country. And when you come to the death, produce causes about 23% of the foodborne illness and the deaths in the country, and meat and poultry is the maximum number of deaths that are coming because of this. But there are still lots of illnesses and deaths that we need to figure out. But this is again data from 1998 to 2008. This is not an exact association between food and pathogen. One of the important challenges for the food safety research was that how do we know exactly how to prevent it from the illnesses happening. That's why President Obama enacted this law, which is called as Food Safety Modernization Act, FSMA, in 2011, which mainly focuses on, instead of responding to an outbreak, how do we focus more on prevention of it. Till now, foodborne illnesses have been investigated only if there is some case occurs, then we already know, or we study then and respond to the illness. But this one talks more about how do we prevent it. But for prevention, many of the scientific community, they are saying that to be able to effectively prevent any foodborne illnesses, we need to know what is that risk-based investigation. That means we exactly need to know this is a particular food causing these kind of diseases, or this is a particular pathogen which causes disease and is always associated with these kind of food. That is not exact association, which is very difficult to come up with. The questions like what pairs of foods and microbes present the greatest burden? Is it like campion poultry? That's the one we have to really worry about, or salmonella and egg or peanut butter? So we exactly don't know. And that is difficult to come up with also. The folks at University of Florida at the Emerging Pathogens Institute, they took this task. This is again a surveillance and network collection of data, and then they came up with lots of numbers. Their idea was to rank the pathogens, rank the pathogen and food association, and rank some of the combinations so that we know where we exactly put our efforts on. What they did is that they took the incidence estimates from the CDC data that has been collected from 1998 to 2008, and also the public health impact they assessed based on the numbers. It's a difficult calculation, but they came up with a number called QALY, which is a measure of health-related quality of life. That means the CDC number that we previously showed you, it does not tell that what is the loss of life. That means you did not go to work, or you sued the food company, or so many costs associated with your like insurance, things like that. Those are not covered in that. But this study tried to cover all those which called as a measure of health-related quality of life. That is QALY. So after this study, they ranked. They came up with 3 different ranks. This is a burden they ranked based on the number of pathogens. So what they say is these are the pathogens that can be ranked. They ranked Salmonella as the number 1 pathogen, and you can see here the pathogen Salmonella, Toxoplasma, and these pathogens, they cause nearly 90% of the illnesses, and 14 billion of US dollars is the total cost they estimated. But this talks only about pathogen. These are the pathogens, but it's not always true when you associate with the food. Salmonella, if you associate with poultry, it's not a big problem in poultry. So that's why they said that, okay, let's do a pathogen food combination and rank them. So when they did that, they came up with different numbers where they say campion poultry is the number 1 ranking problem. Similarly, Toxoplasma in pork. There are some data they were very difficult to do it, so they sought an expert opinion because there was no outbreak in Toxoplasma till now has happened. but there are a lot of outbreaks that they could get the information from. Like, for example, listeria in deli meats is a major problem. Listeria is a big problem in so many other foods, but in deli meats, when it really comes, what is the exact food that I should avoid if I am sensitive to listeria or toxins, then deli meats is the one. So that's how it is. And also they came up with the category where they ranked the foods. So they say that poultry is the food which is causing major loss of illnesses and then cost of health burden and things like that. So these are the different rankings again. This is just I'm trying to emphasize on how difficult it is to come up with a exact reason why a food safety illness can be attributed to. Again, this is the latest number from the CDC, which is a quite lot different from the 2011 numbers. This again emphasizes why it is so difficult to come up with an exact number. Here they said that the increase in the illnesses due to Campylobacter are increasing by 14 folds, and even in Vibrio they are increasing by 43%, which is a big problem in many of the foods. And this is again an easy version of explaining the same thing. Here they say that it's not a good news if there is a 14% increase in Campi, so we had to put a lot of our efforts in coming to achieve a target where we can bring down that rate per 100,000 population from 14.3 now to 8.5, which is not an easy task to achieve, especially when there is 30 cases of Campylobacter illnesses go unnoticed or undiagnosed for every illness. Similarly, it is the same challenge for so many other pathogens. So what do we need for an effective food safety management? If you ask an experts at the IFT, Institute of Food Technologies, they say that we need to have a quick and accurate identification of hazards. We have to have a good ranking system by their level of importance. That's again meaning to say that we have to do lots of research for a lot of new association of food and pathogens. And we need to have identification of microbial control approaches. That means what all those interventions that we need to take so that we can effectively control the microbes or their incidence in foods. But there is another big challenge of addressing the emerging hazards. Hazards include physical, chemical, or biological, any of the hazards, but they're emerging. So let's see what are those emerging hazards or risks in food. Whenever we say, in general, when you say emerging, it is a new risk which is in the process of being understood. We don't know much about it, or it's not been ever quantified. It is just we are learning about it. It doesn't mean it is coming from entirely new pathogen. It may be coming from a pathogen which we already know about, but it is doing something new things. For example, we know a lot about Salmonella being in meat or in eggs and like that. But what if a Salmonella enters into produce? That is something new. Salmonella being able to produce biofilm, it is again a new research. Till now, very few reports were there that Salmonella is also able to produce biofilms and survive in produce and other things like that. So those are the kind of things. And the risks that have no track record, which that's again the same thing which I was saying, that there was no evidence of, there was no occurrence of such kind of incidences that have been happening. Those are emerging risks. And risks that are expected to grow greatly in significance. For example, if a pathogen like Salmonella comes into produce, and produce is the major food consumed by so many people, and then it's a big risk. When it comes to food safety risk, emerging food safety risk, it's a new risk emerging to different kinds of foods. Like, for example, a pathogen in poultry getting into produce, pathogen only in produce getting into other meats and things like that. What are those emerging foodborne pathogens? Pathogens that are emerging in foodborne diseases are those that cause illness that have only recently appeared or been recognized in a population. That means in this particular area, this pathogen was never existed. Things like that. When E. coli outbreak happened in 1980s from the Jack-in-the-Box incident, nobody knew about E. coli at that time. But that was an emerging pathogen at that time, and still now it is, because it is showing up in so many different foods every now and then. So that's again emphasizing the point that the E. coli, though we know everything about, they are well recognized, but they are rapidly increasing in their geography and their incidences like that. And these are some of the emerging foodborne pathogens. You might find a lot of familiar names here, but there are some names like Archobacter species, which is not been implicated in food safety till now. But now there are some reports that are coming up in food. The problem with this food is that it's a combination of Campi and Listeria. Listeria is known to grow in a very cold temperature, like in refrigeration temperature, but this pathogen can sustain colder temperature up to 15 degrees centigrade, and that means it can reach out to more different kinds of food. And one of the important character of Campylobacter is that it cannot survive in aerobic condition, but this does. This looks exactly like Campylobacter, but it can tolerate aerobic condition. So it has the Campi character and the Listeria character, so it is becoming a diverging problem in many of the foods. So the reason why pathogens emerge is again a complicated understanding, but there is a lot of connection goes on between the host, the pathogen itself, and the environment they grow in. For example, in case of agent, that is the pathogen factor, its ability to tolerate certain antimicrobials, or its ability to grow in a new environment, or it's evolving itself constantly. That is the agent factor. When it comes to host, for example, if it started showing up in elderly or in very young people, that is a new kind of host they are developing, or the host factors like immune compromised people. So those are the kind of things which will encourage the pathogen to grow in that new environment. And when it comes to environmental changes, like things like climate changes, or change in the weather conditions, or economic development and land use, technological development, and so many other things that will lead to emergence of any pathogen. So it has to be an interaction of host, agent, and the environment altogether. This is some of the examples where new recent emergence of pathogens and diseases have been discovered in all over the globe. It's not restricted to one particular geographical area. But there is no significant, or there is no one particular reason why pathogens emerge, but it has been agreed that many of the reasons that are known, like newly identified host, if the pathogen was not reported in these kind of hosts, that means it is a new pathogen in that host. And known pathogens spreading to new geographical areas or population, that means global existence of the pathogen. And old disease reemergence. There are some diseases which we thought they are extinct, but they have started reemerging because of some of the pathogens. So what are those key issues that we have to look for? What are those key issues that we need to see? One of the thing is, the biggest challenge is globalization of food supply. The peanut butter that we eat today might be coming from anywhere in the globe, or it might have been processed just yesterday. The produce that the fruits that we eat. And other thing that we need to look for is the processing technology and novel foods. There are new foods that we are coming up with. Even organic food itself is very vulnerable for pathogen infection. Those are the kind of challenges that we need to address. And changes in food consumption habits. You might be consuming one certain kind of food, and now you are tasting new kind of food because maybe you moved out of country or something like that. So your body is not adjusting to those kind of things, new kind of food habits. And as at-risk population, people with already pre-existing conditions, or immune compromised people like that, and evolution of the pathogen itself, like its ability to change, and understanding of the consumer, how to have a food safety practices and integrated system. But I will talk about globalization and pathogen evolution to be narrow our focus on one aspect. Global travel of food is another biggest challenge in food safety area, because our food that we eat today comes from so many places in the globe. And when we talk about global travel of the food, a lot of things, discussion that happens is around food mile footprints. There has been estimation from an independent research group, they call it as shrink that footprint. They say that the footprint that we have from the transport of food on water is much less if you compare it to the food that travels via air. That means the food challenge is coming from a lot of imported foods. So what are the food miles and how do they affect our world? Food miles are basically calculated by considering the time it travels and how long the food travels to reach your table. So that's how they come up at that number. And in the US, on an average, they say that the food travels from 1,500 to 2,500 miles. It's very common. You eat strawberries all the season, and they might have been processed in California or somewhere else in the country, and they have traveled thousands of miles. So that's how it is. And the fastest growing source of greenhouse gas emission is the food miles. I'll show you this graph. It will tell you the personal footprint, which is a measure of how a person's lifestyle contributes to the climate change. Here, the carbon dioxide equivalent in tons, you can see here the maximum number of emission that comes from is food. So that means food is one thing which is a big thing to address, even if it is a climate change issue. And we also know that by 2050, there will be a huge growth in population by 34% higher than what we have today. And we will not have food to feed all those people. To be able to feed all those people, we need a land area of this size. And increase urbanization from 49% today is going to go to 70% of urbanization by 2050. That again has a lot of demand in terms of quantity of food that needs to be available to feed these people and things like that. Other important thing is evolution of pathogen. Again, it is the interaction of agent, host, and environmental things. But one of the major contributing factors are the evolution or the selection of particular organism to new environment, and how its ability to adapt to the extreme temperatures or pH or any other growth conditions, and new hosts or vectors, and antimicrobial resistance. So I will come to the antimicrobial resistance in a minute. This is a recently review published in Nature. They talk about how the emerging framework happens in a infectious disease kind of situation. Here, imagine a species jumping from an existing disease to a new host, and there it is acquiring a new characters. Many times we end up thinking that it's a new ability that this pathogen has evolved into, but it might have that particular resistance already inbuilt in that. Only thing is it has never explored that option. But now it has come into a new host, it has changed its entire way of behavior itself. And then that's how it gets a new characters and enters into new area. So this is how the pathogens usually jump from one species to another species. When it comes to antimicrobial use, the Farmers and Ranchers Association, that means they basically talk in favor of antimicrobial use. They say it is just a part of our routine practice. We do the nutrition care or the veterinary care or the proper housing facilities, just like that. Antimicrobial use is one of that, which is not 100% false, because antimicrobials are essential to keep the animals and even people safe. But only the problem starts when the antimicrobial usage gets abused. And we have to be worried about that, because in the US, the antibiotic resistant infections are responsible for an estimated 20 billion in excess healthcare costs, and about 35 billion in social costs, and 8 million additional hospital days. So it is a big significant number, and it's a big food safety threat. Just this information will tell you how the antimicrobial resistance happens, how these organisms acquire antimicrobial resistance. Let's say, for example, in the farm, you feed animals with some antimicrobial infused feed, and the organisms that are present already in the animal, they are constantly exposed to this low dose of antimicrobial, which is safe dose, which is allowed by FDA limits, which is not causing any harm to the organism itself. But because of the continuous and long-term exposure to these antibacterial agents, the bugs that are there in the system becomes resistant to that. You will not be able to kill them. So then the animal excretes those bugs into the waste system, where they again thrive and develop the antimicrobial resistance. But the problem is they again exchange their resistance to the new bugs that are already there in that. So that's called as horizontal transfer of genes. So they exchange these genes to the new organisms, which are again enter into the system where these animals are slaughtered and processed, where the people who process them again come in contact with these antimicrobial resistant bugs. These are not the usual E. coli or Listeria infections. These are the strains that have become much more resistant for antimicrobials. And that's how the antimicrobial resistance enters into the social world. This is an example how the antimicrobial resistance spreads in the whole community, not just by food animals, which is again similar information here. Here, another way is imagine a scenario where he gets infected and hospitalized. He can directly come in contact with other people in the society, or he can transfer that to the another new patients. So this is how antimicrobial resistance, the pathogens that are resistant to antibiotics, spread among the people. And there's nothing much we can do about it because it's very difficult to detect, to know, unless you have been treated with another new dose of antibacterial agent. And these are some of the pathogens that have been already reported to show multiple resistance or developing new resistance for antibacterial agents. So what CDC has done is that they said, okay, let's take these are some of the important steps so that we can mitigate this kind of problem. First of all, we need to prevent infection that happening, the new infection that's from happening, and by preventing the spread of these resistance among the organisms. Tracking resistance pattern, that means let's track from the resistance, let's say these kind of resistance is already known to exist in the manure, and see how this resistance keeps moving in the system. And improving use of today's antibiotics. Now you might be in the very latest news that FDA is trying to phase out all the antimicrobial use for animal health in the animal practices. But this is still voluntary right now. This is not a regulation. It is they are encouraging people to voluntarily phase out all the use of antimicrobial agent. That means the antibiotics may be gone, or people may choose not to use them. That may be a good thing in terms of not having a resistance problem. But I don't think that it will solve the problem at all. If we stop using the antibiotics, we don't know what will happen. We may come across new pathogens. There's not much options we have. One of the options we have is to come up with the natural antimicrobials. Dr. Muriana's lab is doing a lot of work in that area, and I will talk about 1 or 2 papers that I have done in that area. To summarize, food safety challenges that I said that it emergence and spread of new microbes and new hosts. That is one of the important key factor when we talk about food safety challenge. And globalization of travel and food supply. That means the food goes, or the people move from country to country, and they carry the bugs everywhere. That's the easy way of spreading the diseases. And the rise of drug-resistant pathogens is again a biggest challenge. And when it comes to managing the emerging pathogens, we said that recognizing is a very important step. We need to understand where the pathogens exist and how do they behave in the environment, and things like that. And investigation should focus on diagnosis and surveillance, and applied and epidemiological and ecological research is the most important thing to focus on. Again, education to the consumer, education to the producers, and things like that. And information, international interdisciplinary interventions. You cannot just be a microbiologist and think that we are done. You have to interact with so many other folks to understand, like for example, epidemiology research and all is not a major focus of microbiologists. But the good news is that 70% of the people think that food is much safer in the country, which is also true, that the food is the safest food in the US compared to any other countries in the world. And from the 5 years of food safety surveillance data, they have seen lots of success in controlling many of the problems that we had in 2009 compared to 2013. One positive note is that there is always a light at the end of the tunnel. Food safety challenge that we have now is not end of the world, but we have to keep moving and keep working towards how to understand that problems. I think we have some more time to talk about some of the research work that I've been doing. I will go very fast on that. This paper talks about natural antimicrobials. That means how do we have, is there any alternative use of antimicrobials that are already existing? Like I said, I showed you a slide where some of the pathogens, Salmonella, Staph aureus, Listeria, E. coli, these are the pathogens that are acquiring resistance for antimicrobial agents. So we wanted to test if we can come up with a new antimicrobials that can be prepared from a very natural source. So here we used a pecan shell. Pecan shell is just a waste commodity now. We visited the pecan shell farmer and asked him, what do you actually do with the pecan shells? He showed the burner that he has next to his facility. They just burn it. It's a very cheap, and in fact he said, I have truckloads of it. If you want, you can take it for free, and I can give it to you every now and then. So it is a very cheap commodity. We wanted to see if we can use that and come up with any antimicrobials. What we did that we took the pecan shells and subjected them to an extraction process. In the first paper, it's an extraction process where we used acetic acid, methanol, and solvents extraction. It is not If you want to address these antimicrobials to an organic producer, it is not an organic kind of extraction, but it is still a natural process. So we subjected these shells, which are basically dried shells from the pecan, whole pecans, and after extraction, we tested them against several of these pathogens. We did what we call as the minimum inhibitory concentration estimations, which is called MIC. The way we did is that basically you start with the highest concentration of the antimicrobial extract, keep diluting it in a PBS or a buffer till you get the lowest concentration that you want to test on. And this was done in triplicates, and same was done in all these wells. Inoculate with the bugs that you want to test on and leave it overnight. If they grow, they will show up. And take this, mix it very well, take it with the pipette and just do a stab inoculation. That means you just barely inoculate on the agar and then incubate that. If they are resistant, they will grow next day when you see that in the plates. So here you can see the bugs did not grow here in other tests that we're doing. So that means they were much sensitive for the different concentration. So that's how you come up with an effective concentration of the antimicrobial. Here, one thing I forgot to mention is that there are two groups of antimicrobial extraction that we prepared. The first ABCD is water extraction. That means we just use the hot water, boiling water, to extract the antimicrobials from the pecan shells. And EFGH is the solvent extracted antimicrobials. You can see that the antimicrobials that were the solvents were used to extract, they are much more effective. That means it takes a very low concentration of those antimicrobials to be able to reduce the population. That means solvent extraction was a very good way to go in this scenario. And again, here we compared different solvents. We wanted to see what if the solvent itself is killing, not your antimicrobial, because the solvents are much more dangerous. So like we suspected, it really happened that even acetic acid alone kills the bugs, and even methanol itself. So this is again, when we did the solvent extraction, definitely there was much more log reduction, or much more less concentration of the antimicrobial that was required. But still, we were not happy with some of the information. We wanted to know, can we avoid use of solvents? This paper, what it gave us is that solvent extracted antimicrobials that prepared using pecan shells indicated significant difference between their inhibitory concentration. That means it depends which solvent you use for extraction. If you use acetic acid, there's a change in the concentration because the antimicrobial that you extract is much more depends on the solvent that you are using. And the liquid smoke sample tested in this study could serve as a effective natural antimicrobial. That's what we wanted to infer from that. In the follow-up study, we wanted to see, can we do this extraction in a way which is suitable for organic farmers. That means we should not use any of the chemicals in the extraction process. So we came up with an extraction process which is, we are trying to patent that, so I will not go into detail that. But what we did that we extracted these antimicrobials and tested their efficacy and natural on some of these strains. We used Listeria, but this time we used a food system where we took the Listeria pathogen and inoculated them on the chicken skin of the chickens that we took it from a drumstick, and I'll show you. Here you can see that this is the skin from a drumstick. We wrapped it on a stainless steel ring which was autoclaved, and then used a twist tie to keep it in place. And now you dip it in the inoculum and before or after treatment of the antimicrobial and see if it is effective or not. But in between we give some time for the Listeria to adhere onto the skin, and then we treat it with an antimicrobial agent. That was a very simple process, and we use this against several different strains of E. coli that are already known as outbreak strains, and we used a cocktail of these strains. What we did is, here we wanted to know what if you roast the shells and then extract, or if you do not do any roasting and then extract, if there is any difference. Roasting gave us much better results, mainly because roasting evaporated some of the natural moisture in that, and that's how the antimicrobial concentration inherently was much higher concentration in that. First we tested with natural population of natural flora on the skin. So definitely when you treat it, there is a much more reduction in the pathogens, I mean the natural flora. And when you do the treatment with the different timings, you can see 15 minutes was much more effective than 30 minutes, which was strange, but I kept on getting the same result. And so it wanted to be effective in 15 minutes. I think maybe the antimicrobial is losing its efficacy in that time. And we also wanted to see what if we use the what if you inoculate the organism first and then treat, or you treat the food first and then see if pathogen comes in contact, survives or not. So nothing much difference was there whether you treat it before or after. The effect was pretty similar. This is again the same result where you can see the difference between shell extract or the shell powder extract. When you do the powder and roast it and before extraction, you will see a much better result. That means roasting and extracting is a good option. Again here the same information, where the difference here is the first and second fraction. In the extraction process, I took the fraction very early in the extraction step and again very later in the extraction step. There was much difference in the first fraction and second fraction. I think the antimicrobial effectiveness was much higher in the first fraction itself. So that means it will save time so that we don't have to do the all over extraction or thorough extraction. Just a 15-minute extraction itself will give you the effective antimicrobials. These are the things that will be important when you really want to commercialize or scale up your production. So this is the same repetition of information, but here what we learned was that this was prepared from an organic pecan farmer. The shells that are collected from organic pecan farmer, that means these are qualified as an organic antimicrobials. We did not use any chemicals in that. And to extend that further, we wanted to see what if we do a combination of antimicrobials against different pathogens. And we wrote a grant for USDA. Luckily, we got funded from that, and this work is going on. These works were done when I was a postdoc in the University of Arkansas, and Dr. Phil Crandall is managing that work. Other work that I did with my graduate student was about public health importance, where you might have seen in restaurant that normally they use wiping cloths to clean the table. We asked the question, how effective are those cloths? Because one of the food safety challenge that we are encountering now is that till now we have been using a sanitizing agent which is called as quaternary ammonia. A lot of pathogens are not responding to that. That means they are becoming resistant to that. We wanted to see if there is any combination of antimicrobials that we can come up with, and what is the best cloth that should be used in the restaurants. This is the importance for the public health point of view. Here, cross-contamination is the buzzword, where we get contamination from so many places, especially in the restaurants. 80% of the reported foodborne outbreaks that have happened because of the contamination people get from restaurants. So that is a big issue. That's why we wanted to see if we can come up with something in this area. And we know that a lot of food service facilities, they routinely do the cleaning practice, but we wanted to know how effective is the practice that they do. So in the university cafeteria that we contacted, they said that they use different cloths depending on the availability or depending on what their manager gets. So they use 4 different cloths, which is a terry cotton cloth or cellulose blended cloth and all that. And there was a company called Ecolab. They wanted us to test this sanitizing agent, which is called as SDC, silver dihydrogen citrate. It is basically a nanotechnology kind of compound. They have infused silver ions in their agent. So we wanted to see if it is effective. But we wanted to know if we can detect the contamination then and there itself in the real time, not taking the sample, going to the lab, do the estimation, which is again going to take more time. So we wanted to know, can we just swab it on the table right there and see if it is clean or not? So we wanted to focus on just whether it is clean or not in terms of bacterial load or anything like that. So we took a technique that is called as ATP bioluminescence assay. This is a handheld device which looks like a pH meter, which is made by System Sure Plus company. What it does is it works based on the bioluminescence principle, where there is an enzyme called luciferase. You must have seen these bugs that glow. We call it glow bugs. How they do it is they have an enzyme, luciferase enzyme. When it comes with the heat, it produces the luminescence, bioluminescence. So this instrument is prepared with the same principle. Here in this upper device they have this enzyme, luciferase enzyme, and this is a cotton swab where they use this to swab the area like here. This is what exactly we did. We took a 100 centimeter square area and we prepared this metal sampling area so that we can restrict ourselves in that area. There is a particular way how you should sample when you do this kind of sampling. This is called a zigzag sampling from both directions. That's exactly what we did here with the cotton swab. Put that cotton swab in the device and then break this tip off. That will release the enzyme into the buffer, and then shake it for 15 seconds and then read it with the system. It will show a reading which we call as the RLU, that is the relative log units, which will tell whether the system is, the table surface is clean or not. If the reading is more than 30, it is dirtiest. That means very dirty area. If the reading is between 11 to 29, you have to watch for it. You have to clean more. And if the reading is less than 10, that means it's a clean area. So this will tell for if you are a restaurant owner, if you want to know whether the cleaning is happening or not, you just have to do this. In 15-20 seconds, you will get the result. It will tell you whether your table is clean or not. But the question is, can it be reliable? Can we use it as a reliable estimate? Can we compare it with the total count of bacteria? If we do a bacterial count on the table, can this be compared? It's very difficult to compare. I don't encourage you to compare both these. These are entirely two different things, but you have to use this method only to estimate whether it is clean or not. And we did this. What we did is we asked the University Delhi folks to clean by their own method, because if we go and clean, there may be a lot of variation that we introduce. We gave these 4 cleaning cloths to the folks that they regularly clean, and asked them to use 1 cloth at a time for 3 weeks at a time. And we every time we went and collected samples. We took about 360 samples from 4 different cloths, and we saw that based on the ATP reading, these are the 2 cloths that showed the very lowest number. The RLU was converted as a log number here, and the non-oven cloth, which is widely used in the restaurants, was not effective at all. Now we wanted to see what if we combine the cloth with the sanitizing agent that company wanted us to test. We'll tell them, okay, if you use this sanitizing agent with your cloth, and by the way, this here was done with quaternary ammonium. That's a very common antimicrobial that people use in the restaurants. So those are not effective at all. So that's why we wanted them to test with this agent, the sanitizing agent. Here, these are the ATP readings again. You can see that dairy cloth, if you use just, if you don't use any cloth at all, this is the number. And if you use only the cloth, you can reduce a little bit of contamination or the dirt in that. But if you use with the sanitizing agent, you can be very effective cleaning. So that's what happened with all other cloths also. So that means no cloths differed significantly from each other when you do this practice. That means all the cloths were good enough. Here, we asked the question, what if we take the bacterial counts and compare with the cleaning method? So exactly we did a different table, of course. We cleaned the tables with the cloths and sanitizers and took the swab and did the microbial enumeration. It compared very well with the ATP bioemissions monitoring system. That means we can rely on that system and understand that it is doing a very good cleaning. So that's again the same thing, that ATP measurements can be relied, but you cannot replace the total plate count method, and it can give a real-time monitoring of the cleanliness in the restaurant kind of facilities. This is a different paper. I don't have much to talk about this one. And coming to my 2 points, just one more slide. What work and now I'm focusing is on, since I'm in a pharmacy college, the students that come to my lab is all pharmacy background. They talk only about health and human health kind of. I cannot talk about a lot of food safety areas. So we are working on, we started working on this influence of dietary choline. There was a recently paper from Harvard University. They said that people who eat just 1 egg a day have a higher risk of heart disease. As a food scientist, I really don't believe that. I think it might be just a short-term peak that they noticed, and they published these big papers. We really want to see if egg consumption, like choline is the major nutrient in the egg, if you give choline to the mice and then see if they can increase certain genes that will metabolize the choline and form into product that cause heart disease. But another important thing we are testing is there any intestinal bacteria playing a role in that? That means how your metabolism is working. If you take probiotic bacteria regularly, can you mitigate that kind of risk? And again, there is a lot of work that has been done on toxicity, liver toxicity in our university campus. And I wanted to test if green tea extract or milk thistle can help in mitigating that kind of liver toxicity. There is a lot of liver toxicity problem with acetaminophen that a lot of people take. There is a lot of emergency visits from this abuse of this drug, but not much treatments are there in that. So we want to work in that area also. With that, I will end my presentation here. If there is any questions, feel free to ask me.